Shooting it raw?
Yes. Shooting it raw. Hello, everyone. For this week, I'm going to do a reissue of the episode I recorded with Anna Blocki. And this was in December of 2021. And I think it's a great one. It's a great one for learning about tissue engineering and things called an extracellular matrix. So yeah, it's a good one, and hope you enjoy and are inspired, because we need more scientists, and Anna is an amazing, amazing researcher. So yeah, here's to you. Anna Blocki, yes?
Yes.
Yes. How do I say your last name?
Well, it depends on in which language.
Okay, how do I say your last name in French?
No, I mean like either German or Polish.
Okay, how do I say it in German?
Blocki.
Blocki. Mm-hmm. And how do I say it in Polish?
Błocka.
Oh, Błocka. And so what do people call you here?
They use my German name because I entered Hong Kong on a German passport. So officially for Hong Kong, I'm officially just German.
Okay.
Yeah.
Okay. And so again, in Polish, it's what?
Błocka.
Błocka.
Mm-hmm.
Anna Maria Błocka.
Anna Maria Błocka.
How do I describe what your profession is? Like, what do you call yourself? Like, bioengineer? What?
I'm a tissue engineer.
Tissue engineer.
Yeah. Well, So basically, I do work in tissue engineering, do research on tissue engineering and regenerative medicine, and both fields are very overlapping. So sometimes you cannot really make the difference in both.
Right. I don't know your work. I try to figure out, like, what... Are you a molecular scientist? A geneticist? No, like none of
Yeah, no, it's very interdisciplinary. So my undergrads were in molecular biology, so I'm a biologist by training, but my PhD was in biomedical engineering.
Okay.
So, and as you go along, it's not only what you learn in terms of education, but basically what you're working in. So you take things from biology, medicine, physical sciences, engineering, and just, you mix and match as you go. Uh-huh. And in a way that's one of the amazing things, because if you don't know something, you either get to know it, so you learn. So basically you never stop studying, or you get someone who can do it for you.
Right.
So then you go into collaboration.
Okay.
Yeah.
And you got your undergrad where?
In Germany at the University of Recklinghausen.
And then you went
I went for a PhD to Singapore afterwards.
Singapore. Yeah. Okay. We are looking at an image that... Okay, let me describe it. So, in terms...
That's going to be interesting.
Okay, yeah, so it's not a photo... Well, it's a graphic. It's not... Is it strict? It's not a photograph. It's a rendering.
No, I mean, it's a photograph.
It's a photo?
Yeah.
At 10 micrometers?
No, that's the scale. So this bit, which is like, I don't know, a thumbstick, is basically the length of 10 micrometers.
Okay, how long is 10 micrometers?
It's 1/100th.
Assume I'm an idiot. 1/100th?
1/100th of a millimeter.
1/100th of a millimeter. So, okay, like you can still kind of picture it. Okay, so the image is... It's in square format. It is, it's, imagine looking at a very kind of 3D spider web where the spider is deep lost in a cone, but the webbing is yellow with some green, and there's purple, and I assume these are all colorized. It's a colorized graphic. It's not actually light that we're looking at.
Yeah, I mean, it's something which the software describes with a certain signal.
Okay.
But the signal had a specific wavelength.
Right.
But if you would look directly, I mean, like, this is a microscopy picture, obviously, right? Yeah. So you could actually see the colors by eye. So they're in the visible light range.
So what would it just look, since it's tissue, I mean, okay, because of what you are professionally, I assume this is some kind of tissue. It looks like fibrous. Okay, so in between the yellow and the green and the purple, there's like kind of black. I guess the purple is space as well, but the black is the kind of negative space, I guess. And then, so there's this like, it's like looking at a sponge, I guess. And so why would you call this a portrait of your mother? No, clearly not. No, I'm an idiot. No. Of course not. It's not a portrait of anything. It's okay. So what is this? What is it? What am I looking at?
These are actually two cells, which are stained. So the blue is actually the cell nucleus. Okay. So the DNA is stained in blue.
Okay.
And that's actually what is the cell, and what you see else is actually outside the cell. So, so the black spaces are actually not empty; they're just not stained, but everything is like super full. And this picture was stained for fibronectin and hyaluronic acid, and these are two extracellular matrix components. So what you find as a scaffold around the cells.
Okay.
So this basically shows that if you have cells in a tissue, in a petri dish, they are making their own scaffold.
Okay.
So, and this was a very, I like the picture for two reasons. The first one, it is extremely pretty.
Yeah, it is.
And you can basically see like how nature creates things which are just beautiful. It is a piece of art in a way.
Yep.
And on the other hand, this image was a piece of cake, and actually it's the wrong staining.
Okay.
So,
wait, when you say it's a piece of cake?
Well, in a way, so when we tried to publish this picture as a part of a manuscript, we got a reviewer who didn't believe that our red staining, which is the hyaluronic acid, is going to be actually hyaluronic acid. So he was doubting that. And he was right in doing that. Because, so what this is we have fluorescently labeled antibodies which bind to specific antigens. So one of them is fibronectin, the other one is supposedly hyaluronic acid. And we get those commercially. And it turned out that the antibody for hyaluronic acid was actually staining something else and not hyaluronic acid. And we built like a whole storyline over this, because it was about like what kind of role hyaluronic acid plays in fibronectin fibrogenesis, so which is basically the making of fibronectin fibers, which you need to make a scaffold, like this network. And in the end, we were staining something which was not hyaluronic acid. So there was like a lot of work and time and research money going into it, something which actually was not 100% true.
Right. Okay. Okay, so looking at it now, I can see that there is some reddish stain, which I maybe didn't mention, closer to the blue. Now, the blue is the cell, or the blue is the nucleus?
The blue is just the nucleus.
Wow, okay. But so you can see the red, and you can sort of see yellow and the bleeding into the green and all this stuff. Now, assuming I don't understand what you're talking about, which I don't, what's interesting here is that you're making the scaffolding, which is basically the structure that is forming around the cells, or the cells are making it. Like, why is what's happening here of interest?
So normally, so there are two things about the extracellular matrix, and I'm really fascinated with the extracellular matrix. So one thing is, when people think about tissue, they just think about the cells. But especially if you go in tissues like, for example, the bone, there are actually not that many cells inside. It's mainly just matrix. So matrix actually makes up a major part of the total body. Actually, collagen 1 specifically is the major protein component of the human body, and it's not inside the cell, it's outside of the cell.
Okay.
And you can think of it like if you have a human and a jellyfish in water, you know, they're happy. Once you take them out of the water, you put them on the beach, the human would walk away. The jellyfish will not. It's going to be like all flat and floppy because it doesn't have the skeleton to support it. So the extracellular matrix is like the skeleton of the tissue.
Okay.
So in a way.
Got it.
But it's not only this, it's basically, it has all the signals. So it's kind of like, the cells make it, and it's kind of like an encrypted message. So if another cells come in, it can actually read. And it's not only the biochemical components, so the factors, but it's also the way how they're arranged, so the topography, and also if the extracellular matrix is stiff or soft, so even the mechanical properties will affect how the cells read. So you can use the extracellular matrix to guide biological processes, such as, for example, healing or regeneration. So we are making biomaterials out of this extracellular matrix, and we are trying to kind of encode specific information in it, which will then promote, for example, or regeneration.
For example, blood
Growth, blood vessel growth. Yeah, like revascularization, basically.
Oh, okay.
That's a big topic.
So, is this part? If this is part of your research, you're just trying to understand the mechanics in order to lead to application, or you're actually trying to develop an application?
I mean, like, we have several projects, and this was to understand what role hyaluronic acid would play in the assembly of the extracellular matrix. Hyaluronic acid is also extremely bioactive. It was actually shown that if you go into like some type of fish, like the zebrafish, or into lizards, which can regenerate, for example, the heart completely, right, which humans cannot, is that hyaluronic acid is one of the major components which plays a role there. So if you remove it, instead of regenerating, for example, the zebrafish will have a scar in their heart. So things like that. So there are a lot to it, and we wanted to know in how far hyaluronic acid will affect the extracellular matrix assembly.
Right.
So this picture, in the end, was not part of the final publication anymore, but our hypothesis was not wrong. So hyaluronic acid is indeed needed for fibronectin to form fibers, and it's like, the fibronectin goes first and then all the other components follow. So if the fibronectin fibrogenesis is disrupted, nothing else can be built on top. So that was one of the things.
Wow. Okay. Just trying to wrap my head around it. Okay, so within your team, like, how large of a team is it?
Oh, that depends if we're in the summer or not, because I do have some summer students as well.
Okay. So it's through university.
Yeah. So right now I think I have eight, 9.
Okay.
It always comes and goes.
Shifts. Yeah, for sure. Considering that you're like a total nerd weirdo, right?
Thanks.
You're welcome.
Thanks so much.
No, you're welcome. It's the biggest compliment, because you're using your head to do really fascinating work. Like, how do you listen to, like, somebody who's listening to you who is in their journey? So, for example, in high school, where this is like years ahead in terms of sophistication, understanding, and being able to separate ideas. Like, how do you connect your life or some kid who's developing and making decisions as they go? Like, why did you end up in this field of all other fields? Like, it's pretty esoteric. It's pretty
esoteric.
It's not random. It's really specific, but it's also it's unusual. You're a weirdo.
Thanks so much.
You're welcome. It's a compliment.
In a way, this was always something I wanted to do. I always wanted to go into biomedical research.
Oh, wow. Okay.
And I thought I have to do medicine for that. But I did an internship in a hospital for like 2 weeks, where I followed like doctors through like different different stages. I was in the ICU, diabetes, and so.
How old were you at the time?
I think like 16 or 17.
Okay.
And I saw things which I didn't want to see.
Yeah.
And I also realized that the doctors, they really don't have such a big understanding on like the molecular background.
Sure.
So I think it depends, because there are some really good doctors as well, right? So I do actually have some collaborators who are doctors. I'm not saying they don't know what they're doing, but for them it's more like, okay, so this is the symptom, which means this disease and this treatment. But it doesn't mean that, they are so much interested in the background of the disease in a way. And there's also a lot of, I would say, involuntary contact with a lot of strangers.
Okay.
So
okay, this is it. That's a strange sentence to hear. Okay.
But I mean, like, especially like if you work with patients, you have close contact, you touch them, like. I felt that the doctors are really getting cold towards the patients, which maybe, especially if you're in the ICU, you have to do. Otherwise, you just get depressed at some point of time. But
sure.
And in a way, they sometimes weren't really treating them like as humans anymore. Right. And I really didn't like that. Okay. And then with the combination that I was in the room, people were exposed, I was just standing there. I was feeling awkward. I was I shouldn't be standing here. This is private, you know. Plus that it actually did not really cover my field of research interest. So after I got out of the hospital, I was like, okay, it's not medicine.
That's not you.
It's definitely not me. And but I really wanted to do biomedical research.
Wow.
And then I got into an internship doing like cancer research. And there was a really nice professor who basically sat down with me and explained for hours, like, you know, what kind of genetic mutations there are, what they're working on. That was
just
fascinating.
The context.
And then of course I also did some laboratory work, and I like that too. So I do like working my hands. And but that kind of got me hooked. So I didn't go into cancer, obviously, but I don't know. At some point I realized during my undergrads that my heart is beating for stem cells. So I went into stem cell research.
Nice.
Yeah.
Foreshadowing maybe? Or is that where we work in? Stem cells for heart?
Yeah, well, I'm not doing so much heart anymore.
Okay.
But there are adult stem cells, which are called mesenchymal stem cells, which have a lot of really good properties. So, for example, they modulate your immune system. And you think like if people think about the immune system, they always think about, well, it's to deflect diseases or parasite bacteria. But the immune system is basically like a police force regulating everything, like traffic police.
It's always on.
Right? So they regulate a lot of tissue processes and do a lot of the communication. So in a way, by regulating your especially the inflammatory response, you can decide if something is going to heal or not.
Right. You
know, you need inflammation. Without inflammation, there's no healing. However, if the inflammation is wrong or too long, there's no healing either. Exactly. So it's really about this fine tuning, and those stem cells were actually shown to really be able to fine tune the immune system and thereby modulate healing responses and even promote regenerative responses. So people then worked a lot putting these cells into all different places. And but the thing is, you know, normally you put the cells into a diseased area, right? Which can be ischemic, necrotic. So if something is killing the tissue, if you put in cells inside, they are not going to fare better. So they are going to die. So the idea was, instead of using the cells directly, to use what they are making, which actually has this effect. So people have been working on their secretome, which is basically like all the soluble factors, on exosomes, which are like little microvesicles, which contain like a lot of also signaling factors. And I decided to go for the extracellular matrix.
Got it. I understood what you said.
That's good.
Yeah. Shall we go to the next photo?
Yeah.
Okay. A very arresting photograph. Did you make this photo yourself?
Yes.
You did. There is what is the specific name of the beaker?
It's a beaker. It's just a glass beaker.
Yeah, it's a glass beaker. Okay, so so in this Pyrex, I assume, it's obviously in a laboratory. It's a close-up photo of do you know the species of the mouse?
That's a rat.
Of the rat?
It's a rat.
Yeah, but you know the species of the rat?
It's a Vista rat.
Okay. Vista?
Vista,
okay. It's in a beaker. It's being weighed, I'm assuming, because it's on a weight scale. It's being weighed. Does it suffer from albinism, or happens to be?
It's just the type of rat.
Okay, so it's all white.
Yes.
Pink nose, pink ears. We shouldn't get too attached to it. Yeah. Very, very. So what's arresting? First, as an image, it's okay. So when I had Nushin Mubasiri, who is the artist from Iran, she always puts her face looking at the viewer. And she said that's a way that I engage aesthetically with the viewer. And so in this case, this rat is stuck in this beaker. It can't climb out.
Oh,
but
it could.
It could? Okay. It just doesn't want to. And it's just So it's sitting there. It's and looking up at the photographer, probably wondering, what's a camera? Just looking with a very neutral expression. What makes the image is its eyes are super, super red.
Yes.
And look, I know I'm being really cheeky by adding all of this very soft personification, but it is in a laboratory.
Yes.
And let's find out why and how this rat is so important.
Well, it was one of my first research animals. I got attached to it, which is not really good. So when we eventually had to sacrifice it, I was crying so badly. So this was part of my first postdoctoral appointment.
Okay.
And that was more relying on animal experiments. So it was all about imaging, about MRI imaging, about PET scanning, although mainly MRI imaging. And I was supposed to find a way. So again, I was working with those mesenchymal stem cells, and we were trying to transplant them into heart attacks. And the thing is, the heart attack is basically a dying tissue. So if you put cells in there, they're just not going to stick around.
Right.
So especially since, this is, and if you inject them around the infarct, the tissue is contracting, right? So basically it's just squeezing the cells out. So there's basically nothing left. So my goal was to find a way how to make the cells stay in there.
Right. Okay.
So
now, let me interrupt for just a second. So one of the words that got lost, I think it's amazing that, so your mother tongue is Polish.
Yes.
And then German on top of that.
Polish was basically something I did talk mainly at home.
Okay.
And I've moved away from home a really long time ago.
Sure. Well, so I know this is really taking a sharp left from the conversation, but I also like to look at the person who's speaking to kind of really understand like your personal development and journey. It's like, because here we are, we're speaking in English, and your English is perfect. Somebody will be like, oh, but I have an accent. It's like, we all have accents. It doesn't matter. Your English is actually, obviously super high level. Just every once in a while, I think for somebody who's listening, where the terminology is new, they may lose out a little bit. So you said, use the word infarct.
Yeah. So it's a heart attack.
Okay.
Limited blood supply to the heart tissue, to the heart muscle tissue, which then, because you can think about it like it's a really high performance muscle. So it needs a lot of oxygen, nutrients to actually do its job. Yeah. So it's very sensitive if it gets deprived of oxygen. Right. So, and it dies immediately.
Oh, really?
Yes. So, but then the initial infarct, so which is basically the area of that tissue, is very small. What happens is that this causes a very strong inflammatory response.
Yep.
Which affects the surrounding tissue, which is at risk. And it is also depending on how large this initial infarct size is. The surrounding tissue has to kind of work now also and make up for the lost tissue. So they actually have to work harder. So what happens over time in an infarct is that it actually expands. Okay. Until it can actually lead to heart failure. So patients very often do not die immediately from infarct, but from like the long-term effects coming from there. And again, it's like the inflammatory response. So ideally, if you put stem cells in there, they might not be able to make new cardiac tissue. But if the initial infarct size is not so big, that's fine, as long as it doesn't expand. So it was, in a way, the idea to have the cells there so they regulate the inflammatory response and therefore, stop the infarct size from increasing. Right. Yeah.
Okay, so this particular rat at the moment, what was the study that you guys were doing?
So this rat actually had a heart attack.
Okay.
And you would be amazed because at some point, obviously, these animals had to be sacrificed and then we had to isolate the hearts and we did histology. So we actually looked into like slices and really looked on the cellular level what's happening there. And some of those infarcts were really huge. Like you could never have a human being running around with such an infarct. And these rats were like happily running around, having fun. So they can actually take much more than a human being. Yeah. But basically after because you induce the infarct, you actually have to do a surgery through the ribs. So the third and fourth intercostal space. So you kind of do like a rib spreading and then you go and
this is while it's alive though.
Yes. So it's like a proper surgery with like everything. So, however, later on, when they are just recovering, they're of course on like heavy painkillers, etc., antibiotics. So basically, they're like pretty I don't know. I'm not sure if they're really happy afterwards, but they're definitely, pretty sedated. But still, you try to be careful because since they do have this injury on the rid, you wouldn't just pick them up by the tail, because it stretches them. So I taught the rats actually to climb into the beaker.
Okay.
And in that way, they were just sitting comfortably, and I was like taking them around the lab. And injecting them and weighing them so.
And this particular study was, what were you looking at? What were you specifically looking at?
So this is shortly after surgery. So you can, it's actually, the rat looks happy. So you can see if an animal is in distress and this one is not. So I think I was preparing for, it was either after surgery care or preparing for the next MRI. Because I was doing, the cells which I had were actually labeled with nanoparticles which are visible in the MRI. So I was tracking the cells in the heart of the rat over like 6 weeks.
Okay. So let me just say that again so that I share that I got what you're saying. So you used specific nanoparticles that you injected into the rat, into the tissue.
No. The particles are incubated with the cells, with stem cells, and they take it up.
Right.
And then they're kind of stuck with it.
Got it.
And then you go into surgery, and you induce an infarct, and then you inject those cells. And then they're supposedly inside. And then you try to monitor how long they will stay inside and if your approach to prolong their stay there actually works or not.
And you did this using an MRI?
Yeah.
How did you get a rat into an MRI? You're sedated, I guess, to not move.
Yes.
How large is the MRI? Is it like a little mini MRI?
No, it's pretty big actually, because it's a stronger magnet. You need a much higher resolution than of a human, right? Because the rat is much smaller. So the magnetic field has to be stronger. So I think this was a 7 Tesla.
So a 7. Now I know a little bit a very, very little bit. So a 7 Tesla. Now people are going to hear Tesla and they go, Tesla, the car? What are you talking? What's a Tesla?
Exactly. It's the unit for magnetic strength, of the field, the strength of the magnetic field. So the stronger it is, the higher the resolution of the MRI will be. Right.
And the higher the voltage, the power.
Yes, of course.
And this was where?
In Singapore.
Yeah. In the lab, in the hospital, like where
in the lab. No, because so the thing is that the MRI by itself is pretty large.
Yeah.
But the hole where you put in the rat or the mouse is actually quite small. You just put the rat on a cradle, and then it kind of drives in. And you basically don't want so much distance between the coil, the magnetic coil, and the animal, because that will just decrease the resolution. So you want everything to be pretty close by.
Have you ever had an MRI?
No, but I was present when other people had theirs.
Oh, okay.
And I mean, like you basically, you're also there when the rats are there.
You're not in the room, are you?
Not during the actual scanning because that's really loud.
Yeah.
But you're basically sitting behind the glass window. Sure. And that's for the MRI, the magnetic field is always there. Yeah. Right? So you have to be careful when you walk in that you don't have any metal objects on you, etc., etc. On the other hand, at some point you do get a bit relaxed. So for example, if you go into a hospital setting, everybody will always ask you to take off jewelry. Yeah. For example, I didn't do that because silver, gold is actually, not magnetic. So it's fine. Magnetic,
that's a mistake you only need to make once, I think. And then you're just like, I'll never make that mistake again.
No, but that's fine because, or like once I, for example, had like this little hairpins in my hair and I didn't realize. And I went into the room and I felt the tug.
Wow.
Right? So I actually went out and took them out.
Wow.
But,
Okay. Anything else you want to say about this little fella?
No, it's just...
How long was the study?
6 weeks.
Oh, that's quick.
Well, it's like if you don't do anything, I lost track of the cells after 3 days.
Okay. Oh, okay.
So they were 2 days. So I did one scan 2 days after surgery and then I could still detect them. And any time point later, I couldn't detect the cells anymore. Okay. So what we did there is then we put those cells into hydrogel capsules, which were like a bit larger, ones of 100 micrometers, and injected those capsules inside, and they like physically immobilized the cells inside the tissue. And after 6 weeks, I could still detect the cells by MRI. And then I got the hearts and also showed that the cells were there.
Right. Wow.
Yeah.
Shall we move on?
Yeah, we can move on.
Love it. Love it. In this image, we have 2, 4, 6 human test subjects.
So you know.
No. So it's a photo inside an office space of some sort. It's a photo of a group of people. There, you are within this small group. People are happy, posing for the camera. I can sort of see the logo for Chinese U somewhere in the background there.
Yes. The
lighting's very nice. Everybody looks very pretty, very happy. What is this? Please.
So this is actually an event where one of my students presented her research. So that's actually Joyce. She's in the front. And she won the first prize for the poster, which is hanging in the back.
Nice.
So this was about... So here we were making tissues on a chip.
Okay.
And so this is another research direction, but it's again about blood vessels.
Okay. So wait, tissues on a chip, like a silicon chip?
Yeah.
Okay.
So it's basically now, because I've been doing animal experiments for a while, and first of all, it's something which is not, it's not right to use animals like that if you don't have to in a way, right? And you should try to find ways not to do that. So as I said, like for example, I really got attached to these rats, and I really had a tough time, especially in the beginning, to actually do all these experiments, and especially even see them go eventually, right? So you try to get around that, and one way is to actually make micro tissues, which you can put on a chip. And because you put them on a chip, you can really like monitor them real time, high resolution. And the great thing that they're actually human, because I mentioned that rats can actually take much more than humans. So they are only that representative for like a human response. So very often you do experiments, you go into animals, you think you have it all figured out, you have like a therapy which is going to help you treat cancer or Alzheimer's or whatever. And once you go into a human test subject, nothing works anymore, right? So, also for that reason, an animal model might not be the best way to go. And if you can make those tissues on a chip, you can basically try to avoid animal experiments on a long term, and you can actually have human responses because you can make human tissues.
Right. The chip, like what is the interface? Like what is...
Basically it's a very thin glass slide. And then you have a silicone like cover on it, and the silicone cover has channels or like holes, and you can, for example, then seed cells in there, and the cells will not only become cells, but they also make the exocell matrix. So and then you give them different stimuli, so they do things for you, like differentiate into heart cells or differentiate into pancreatic cells, or in my case, actually go from like being randomly distributed to making hollow vessels, which can be perfused and which behave very, very closely to a normal vessel.
Wow. How fast? Like, what's the time? Because this is part of your work. You have a frame of reference for all of this, whereas I don't have a frame of reference. So, how fast do these things happen in terms of you have this glass slide with this silicon wafer thing with these holes or channels or whatever, and how fast does the action or the sort of the event take?
So the chips, you can make them like in one day.
Okay.
And you can make like several of them. So my students basically have like a little chip factory, and they just have like a huge storage of those ready for use. And then, we implant the cells inside. So that takes, it depends on the student. No, some are really fast.
Of course.
But I would say like, half a day max, depending also on how many devices you're making. Like one device will take you maybe like an hour or so. And then you wait for 2 days, and after 2 days you have vessels.
Oh, wow. So you look like a very young teacher.
Oh yeah, sometimes people think I'm a PhD student.
Yeah, exactly. Yeah, you look like a very
young... That's really nice.
Okay, so from my work through service leadership or service network leadership, which is always about looking at the network of people. So when I see this, where say the output or the focal, sort of the intellectual focal point is the poster behind you guys,
yes,
where she won that award for. She's the author or the creator of that poster, or the one who wins that award. But you're within a group, right? So there's the dynamic of you are the elder. You are the guide. No, but in a network though, it's like different people have different roles. And why don't you talk about what it means to you for you to be in that role?
Yeah. Well, so the first thing, the student in the front, she's the one who was the first author. She's actually, she was a summer student.
With the black shirt or with the white shirt?
With the black shirt.
Okay.
The one who looks like really happy,
who's giving the two fingers.
Yeah.
Not the British V, but the Asian V.
Yeah.
The British one is a little bit ruder.
Right. But you can see also from the author list in the back of the picture, she's not the only author.
Sure.
So basically, so you can see the girl on my right.
Yeah.
She is my PhD student, and she was basically mentoring her as well. So this is really a group effort.
Yeah. Yeah.
And what you can see, although only Joyce was presenting, half of the lab actually showed up to support her. Right. So it's really a great group dynamics that everybody is kind of cheering for the other persons, and people are supportive and working together. And all these successes of this one single person, which is amazing. I mean, Joyce really did an amazing job. Sure. Like her research work was good. She also presented very well, very professional, but it's not only her success, because a lot of people were involved, and it's also their success. So obviously you can see Christy's also very happy because she was mentoring her. Yeah. So she's proud also of herself that she got her that far. And I think this is what I'm also doing. So I really this is one of the fun things in my job, when you see people grow, people being successful, achieving something. Yeah. It doesn't have to be something which is recognized to the outside. It can be also something I'm realizing by myself. So very funny is when PhD students normally first start, they kind of have this, okay, so what do I have to do to get a PhD? Oh. And I was like, no, that's not how it goes, right? So then I give them a year where they scramble to kind of find their way in this whole maze of potential experiments, and they also have to read up on the project. So there's so much information, and which I also showed in the first pictures, many of those informations are actually not right. So if we would have gotten any other reviewer, this would have been published, and this false information would have been out there. So actually 50% of research is non-reproducible.
Okay, yeah.
So for someone who starts doing research, it's very hard to distinguish.
Right, sure.
And then they start doing experiments, and most of the time things do not work, especially in the beginning. They just don't have so much training, so there are technical errors, right?
Yeah,
exactly. Yeah. So it's very frustrating. And at some point, things start working out, and then they become very happy. So it's this, all these failures, and suddenly one thing comes up, and they get really happy about it.
Sure.
And if that repeats a couple of times, they get this hunger.
Yeah.
And they want it, right? So at some point, they actually they take ownership of the project. Right. And then it's not what do I have to do to get a PhD, but then this is, okay, I have all these ideas and I want to get this project. I want to this, So you see this personal development from just doing research as a mean to an end. which is basically to graduate and get like a better paid job. Although academia is not always the best paid options, but I'm not going to complain. But it's more about that they kind of get into their driver's seat. So this is an amazing development.
Yeah, for sure.
And to see that and to see like their personal growth, or like also in a way when they present, like in the beginning, they might be shy, they might be introverted, you know, they just are nervous. And at some point, they're really keen to share what they found because they are so proud of it.
So how long have you been with this group?
Oh, wow. I've been in Hong Kong for 4 years. Not all of them have been that long. So Kendrick, which is like the one with the white shirt, he joined my lab quite early on as an undergrad student, and he's been like in and out. He did his final year project with me, and he's now at Imperial College.
Oh, wow. Okay.
So Christy has also been with me. She actually started as a research assistant and then eventually became a PhD student. So she's been also with me for like 3 or 4 years now. Then the two girls in the front, they're actually in the school, they're undergrad students in the School of Biomedical Sciences. So they have been there for the summer, and I do have them in some of my lectures though.
Right.
And Owen, which is the one in the front who actually is taking the picture, is a final year student. So he's been with me since May. Okay. And KK is in the back, and he's been with me over 2 years now, 2.5 years. He's also a PhD student. You know, the great thing is, what I realized is that once they came to you, even if it's just like for an internship, they tend to come back eventually.
Right.
It's very funny. Like,
so we talk about in service leadership is that as a service leader, your role is really to attract people, right? So a leader who doesn't have people around them is no leader.
You need somebody to lead.
Yeah. Paul would say somebody like a leader with no followers is just somebody taking a walk. Like you're just, so your ability to attract people is really going to set or define your success. And so that feeds your reputation. I mean, all these different factors bring people to you. And if people keep coming back and staying, that must mean that you're pretty good.
They do like me.
Exactly.
At least this is what they tell me.
Sure. That's awesome. Should we move on to the next photo?
Yes.
Woo! It's another group photograph, and it's in a hallway, and so you've got 2, 4, 6, 8. So 8 people, and you're standing in the back holding what looks like a decapitated white cake with some bleeding fruit on the top.
It's mango.
Oh, it's mango. Okay, great. Yeah. There's a little kid who looks very young, probably about like 6 or 7.
Yes.
Maybe a young researcher. Let's put it that way. 6 or 7. Yeah. So this is a great warm group photo. Why is this important? What am I looking at?
So this was so first, this is actually in the building. This is at the entrance to the lab area. And this was my birthday this year. And I didn't tell anyone. So I was trying to hide the fact that it was my birthday. Yeah. Because, I'm still trying not to get older. See if I can stop that from happening.
Sure.
And at some point, my group actually surprised me with a cake.
That's sweet.
And that was very nice. Also, I was very touched by that. Yeah. That they remembered, because being honest, I don't remember all their birthdays.
Right.
And also just, that they actually took the initiative to celebrate with me. So that was amazing. And but why I chose this picture. So you can actually see there are some people which also were in the other photograph. There's some other people. So especially, the girl on the right side, Zoe.
Yeah.
She just got her first paper accepted, and we are submitting her second one. Okay. And she will definitely have 1 or 2 more. So she's totally rocking her PhD. Amazing. Yeah. And she started off as such a shy girl who in the beginning, I felt that she's going to be pushed around by like everybody else, because like she was not really standing up for herself. And sometimes you have to be fierce, especially there's always like shared facilities, and you have to book, and people are basically taking your slots, and you really have to be able, to stand up for yourself.
Sure.
And so in the beginning she was like super shy,
Yeah,
and you know, and then at some point, which came with this, I'm taking ownership of my project. She started to be also very protective of her project,
Sure.
and also to really like making sure that people do not step into her space. So she became very confident.
Okay.
And yeah, but that's not the reason why I chose the picture.
So why did you choose a photo?
Because like the little girl there is actually my daughter.
Oh, yeah.
So I don't know why, but for some reason, I had to take her with me to work. So she was in my office. And so why I chose this picture is because I realized in my job, there are actually not, especially in the higher position, there are actually not that many women.
Okay.
Maybe, well, Hong Kong is a bit different, I feel. But I've been working in Germany before as well. And in Europe, like the moment you move into a research institute or you are like, in a scientific field or engineering field, they're almost like, there's a huge overproportion of men in these kind of professorship or higher leading position. And I always asked myself why. And by now I do understand why. Definitely, I think it's harder for a woman to actually set foot into such a male-dominated field.
Okay.
Especially because like it's like a closed club sometimes.
Okay.
It doesn't mean it's always the case. Like in Hong Kong, it's amazing.
Yeah.
Like it's very open. I also feel that there are much more women.
Okay.
In research here, and it's very inclusive. It's not exclusive. But in Germany, that was definitely different.
Okay.
And then I realized that then there are like some women in these positions, and those are extremely fierce.
Yeah.
And very often childless.
Okay. Yeah.
So I would say over proportionally, many women who like in this position never have kids.
Sure.
And I think they basically decided to go for the career instead for the family. And I didn't do that. So I have a daughter and I also have a son.
Wow.
And I think you don't have to choose. And I think it's wrong to choose.
Well, that's a tough one, because raising a child is a lot of work.
It is.
It's really hard. It's a lot of work. And I think for a lot of women I think that's a real challenge for women who want to have children but also continue in their careers. At some point along the way, just because of fatigue, of scheduling, of access, whatever it is, they face a choice, right? Like I think about, my wife, who with our daughter, when she was doing her PhD and working full time, it was just like she had there weren't enough hours in the day. And it's not only the will. A lot of these women who have to face this choice, who can't do it, they have the will, but who just can't make it happen, right? So you are able to. It's not in the past, because you're still working, you're still teaching, you're still full on and being a parent.
I think if it's a choice of time, then yes. But some people some women just are afraid.
Sure.
To make this step.
Okay.
In academia, you don't really have like long-term contracts in the beginning, at least, right? So you have the short-term contracts. It's really competitive. So when I, for example, interviewed for my job in Hong Kong, I think I heard the most often sentence I heard from a lot of people: you either go up or you're out.
Right.
So you either advance in your career or you exit or you this career path, right? So, and this is how it's been since my PhD, basically. So under such circumstances, some people just do not dare to have children because there's not this stability. They don't know, am I going to have a job in like 3-5 years, whatever. Yeah. How am I going to support my family? So they kind of seek this stability, and in a way, like to have something where they can put children and a family into. Right. And I have to say that I also had these thoughts, but it kind of just makes you scared.
Right.
And if you make decisions out of fear, most of the time they're not good decisions. So I think that at some point I just let go of this. I have 2 kids. My kids are pretty resilient. I don't need to be a millionaire for them to be happy. For them, the happiest moments are for example, in coming with me to work. So Lucia loves it, you know, like
right,
she's center of attention. Everybody wants to talk to her.
And it's got to be so cool for her to go into this place that is so
yeah, well, we have
impressive.
this rule. She cannot touch anything. It's like, don't touch.
That can kill you. No.
Right. Maybe it's faith in a way that even if this doesn't work out, something else will.
Well, okay.
In a way, like I'm not stupid. I'm going to figure something out.
Well, one of the things that's clear is that you're hardworking, which isn't to say that women who can't do it all aren't hardworking, but you're competitive. Would you describe yourself as competitive?
Yes, I love competition. I love dares. I love challenges.
Yeah,
you know.
Yeah. So you are competitive.
Yes.
So why don't you talk or share about when you're in the situation, you won't necessarily think, oh, wait a second, I'm facing this guy who's being super dismissive or sexist or whatever, and here's a moment for me to push back. Sometimes you're just in that situation, and then you catch yourself and you go like, oh, hold on a second. So have you had really intense experiences where you're not in Hong Kong necessarily, but maybe in Germany, where you said it was really male dominated and really a lot, you know, where the people at the top tended to be male, where you were kind of facing a challenge that was obviously like, okay, hold on, this is fucked up. Like,
yeah.
Okay.
Yeah. So definitely, yeah. It's actually why I left my job in Germany as well.
Okay.
So it was a research institute which was really male dominated. And I mean, like, it's not like only women were targeted. Like the head of the institute was not a very nice person,
sure,
you know, but he kind of took an interest in me. So normally, like, you have still this hierarchy. So there's head of institute, then there's head of department. And then I was a postdoctoral fellow there. But, you know, so I would have to report to the head of department, not to the head of the institute. But the head of the institute sometimes went around the head of the department, which was tricky. And in a way, it's just that everybody in these positions, if it was the head of the institute or the vice, I don't know, what is it called? The person who, if the head of the institute is not around.
No, not the dean.
The vice?
Chancellor, vice chancellor?
No, because it's just a research institute, right? It's not a university.
I have no idea.
So anyway, like the whole management thing was all male, and I don't know. There were sentences like, you know, like I know you have a kid, so if you want to have your contract extended after these 2 years, you better do this and this and this. Because actually I was there I got my own fellowship, so I had my own research project.
Right.
So I had everything planned out. Yeah. I had my milestones, and at some point they told me that I'm not allowed to do this project anymore and I'm supposed to do something else. Huh. Which is not very ethical, and they were like basically like using again this you basically you want to settle down here and you can have like if you do this, you know, you will get a contract, you can live your life here, be a happy mom. And I was like, yeah, it doesn't make it right.
Right.
Or I heard sentences like, oh yeah, because she's good looking and she's a woman, let her present because it will look good on the institute.
Yeah.
When I was once like interviewing for also like a group leading position in Germany, and it was like the head of plastic surgery, which would have been fun because he had a lot of funding and a huge lab. He just basically needed someone to like do the things, right?
Right.
And again, we had this conversation, and he knew I just had my daughter, and he's like, well, if you want, like, I can make sure that you are going to have a comfortable life, so you never have to worry about, like, that your contract doesn't get extended. You can settle down here. You can take care of your kid. You can do whatever you want in my lab, whatever, you know, like whatever research direction, as long as my name appears as a lead author everywhere.
Yeah, exactly. You can do everything as long as I can take the credit. Yeah.
Exactly. And I was just, looking at the guy, and I was like, if he thought he's actually doing me something good for me, because like I can be like this mummy with, you know, and I was just like, yes, let me consider that. Thank you. And I never spoke to this guy again. I didn't even like say no, because I just felt that was so rude.
Sure.
And so there have been like a lot of these kind of situations, and it's just that it's kind of a closed club. So many of these men are obviously older.
Yeah.
So if there's like this young chirpy woman, they just don't take you seriously.
Okay.
They might eventually be like impressed by something what you do, or I've been told almost like, oh, that's a genius idea, but that's kind of it. They don't really would put you in a position where, but having said that, I don't have that in Hong Kong at all.
Right.
So Hong Kong is very different, and I really appreciate Hong Kong for that.
Are you taller than most of your colleagues?
In Hong Kong, yes, I think so.
I imagine that kind of helped.
You think so?
No, listen, I was just kidding. Look, these podcasts go by very quickly. I mean, it's been an hour.
Oh, wow.
Isn't that amazing? Thank you. This has been I love this stuff. This is so great. Thank you so much. Awesome.
It was a pleasure. It's great. Thank you. I do love to talk about my work.
It's awesome. It's
right?
great. Woo!
Shooting it raw?
Yes. Shooting it raw. So is life really a gift? Really? Can you make every second count? That's the whole point of the podcast. So if you like what you've seen and you're inspired, because that really is my mission, then please give it a like, subscribe, and share.
Shooting it raw?
Yes. Shooting it raw.